Hi guys, welcome to your first bioinformatics homework assignment. This assignment and the next 2 weeks are all based on enzymes that are put into laundry detergent to make your clothes cleaner. Now you should have already seen a video that talks about detergents, how they work, and the role that enzymes play in stain removal. Now we're going to start looking at the sequences of those enzymes, and then we're going to look at the sequences of the genes that encode those enzymes. Now, the specific objectives of this homework assignment are to identify 3 different enzymes in Tide laundry detergent, use NCBI to define molecular properties of the enzymes, and then visualize and document 3D structures of detergent enzymes. Now, throughout these bioinformatics assignments, we're going to look at 2 types of biological code: the code that makes up DNA and the code that makes up protein. And you need to distinguish between these 2 when you're looking at these databases. The code of DNA is made up of 4 letters: A for adenine, C cytosine, G for guanine, and T for thymine, which make up the DNA, which encode the genes. The code for RNA is the same code, except in that case uracil is used instead of thymine. Now the code of proteins is made up of 20 letters, which are represented by 20 amino acids that comprise the proteins. And remember that an enzyme is just a type of protein that catalyzes a metabolic function. In most bioinformatics outputs, the amino acids will be represented by the single letter code. But for our assignments, we want you to answer using the 3-letter code because it's really closer to the full amino acid name and it makes more sense. Now you can use this slide in order to interpret the 3-letter code for your assignments, or you can go to this website and get the same information. Now these homework assignments will require you to fill out cells in an Excel spreadsheet. So if you haven't already downloaded the latest Microsoft Office suite to your computer, you need to do that now. The license is free and there's plenty of tutorials on how to use it. All the assignments are on the class Canvas site. So go to the first assignment and download the Excel sheet. You need to complete this table. I'll give you the first column and a few bits of information in the other cells. Across the top of the table are the trade names of 3 enzymes sold by Novozymes. First, find these enzymes in the Novozyme Household Guide catalog, and then complete rows 2 to 5. So this is their list of enzymes that companies can buy to put into laundry detergent or dishwasher detergent or whatever they like. So what I want you to do is look through this list, look for specific trade names that are seen here, and then under the trade name, it explains what the enzyme class is, what the activity, what the reaction products, and what the consumer claims are, or really what the stains are, what the thing's doing in the enzyme. So you can fill out the first couple of rows from that Excel straight from here. The one number that is important is this number right here, which is the EC number. The EC number is the enzyme commission number, and it's an official numeric classification system that describes an enzyme activity. The EC number has 4 digits that are separated by periods, and each represent a finer classification of an enzyme activity. The EC numbers can be important because it's a standard nomenclature that defines the activity of an enzyme, regardless of what it's called. Now, once we know something about the enzyme activity, we can learn more about its molecular structure by going to the National Center for Biotechnology Information, or NCBI. NCBI is a nationally supported center that houses many important biological databases, such as GenBank, PubMed, and also a host of bioinformatics tools that you can use to mine the data that's presented in these large genome databases. Now there's a lot of information at NCBI, and this course will only cover just a small fraction of those. So feel free to explore these on your own. Now if you type a search term into the search box, I'll try protease, NCBI will by default look at all 35 databases that are housed at NCBI. Now let's just go back to the home base and let's search for one of the terms in the homework assignment. So search for the terms that I've given you in bold here. If we search for the accession, this is an accession of a protein. So we'll put this number in the search box, but we're particularly going to search the protein database. So slide down here to see protein, say search. You could also search all databases and then go to the protein results, or you can just key in what database you want to search for immediately from the search box, which is what we did. This comes up, it gives a GenPep view of the protein. We'll change this view later to look at different formats. The GenPep view gives you the size of the protein in amino acids. When the protein was described to GenBank, November 2007, the last update. The definition is the full name, subtillicin Carlsberg. This is recommended name, full recommended name. Now this protein was bioinformatically put together from many different sequences, and to recover all those sequences, that's what's under the DB source. So each of these represents sequences that this composite protein sequence is comprised of. As you scroll down, you can see the keywords. It explains something about the enzyme. It's a serine protease, so we know it's a protease because it cuts protein. Serine is an amino acid inside of a protein. It's one of the amino acids of proteins, and this protease cuts specifically at serines. If you look down to the source, this shows what organism this enzyme comes from. And if you click on that, you'll see a full lineage across the top that talks about this is the genus and the family. And as you look across here, each of those kingdoms will be described. And for your homework assignment, you want to say what super kingdom these enzymes are in. So you can find that by scrolling over. So in this case, it would be bacteria for the Bacillus licheniformis. As you scroll further down, what you'll see are references where the publication of this sequence has been put forward. So this one's in the Journal of Biological Chemistry back in 1968 was the first time this was sequenced. So there's a lot of information about this enzyme. If we look at comments, it explains what the enzyme is about. Subtillicin is the name of the enzyme. It's an extracellular alkaline serine protease. Extracellular means it's secreted, it's released from the cells. It shows it down here again, being secreted. This is important for an industrial enzyme because it's a lot cheaper to process if it secretes the enzyme into the media than it is to try to break the bug apart. It's used as a detergent protease. It's sold under the name of alkylase by Novozymes. We know that. We saw that was the case. As you look down a little further, it'll explain what the gene is that encodes it. We'll look at this next week. The protein, what the protein is called, and the EC number. And again, you've already seen the EC number. But if you click on this EC, that'll tell you what that reaction is that enzyme 3,4,21,62 is doing, and it'll describe what that enzyme is about. The next section talks about features of the protein. In particular regions, it's known what those regions are doing functionally. So as you scroll down, the take home here is that there is a lot known about this enzyme, which you may guess since it's been studied for the last 50 years. As you get to the bottom, you'll find a sequence of the enzyme, and your homework assignment will ask for the first 6 amino acids of that sequence. And later assignments, you're going to be asked to manipulate these sequences. If you just try to copy the sequence direct from here, what you'll find is you'll have these numbers in the sequence, which will screw up any software that's trying to deal with that sequence. So in that case, what you'll need to do is you want to reformat it into a FASTA format. And the FASTA format is a standard format for bioinformatics. It's used both for proteins and for DNA. And it consists of this greater than sign followed by a descriptor of the enzyme or the gene, followed by just the letters, the genetic code of that protein or gene. Now let's go back to the GenPep view of this. What you'll see as you look over to the right is a section called 3D structure. This is where we'll find the 3D structural information about this enzyme. You can get the same information if you click down here, related structures. So either click there or up here. And what you'll find is a series of enzymes that have been crystallized. You want to look at the Excel table and make sure that you're looking at the same PDB ID number. This is the protein database ID number. In this case, it's 4C3V. So go ahead and click on this. That's once you get the right enzyme. This view shows a summary of the enzyme structure. The metadata about the protein, its name, source, organism, are all shown at the top here. The phrase biological unit, monomeric, means that the enzyme is functional as a single peptide chain. Other enzymes you will encounter may require 2 or more copies of the protein in order to be functional. If it requires 2 proteins, it's called dimeric. If it requires 4 proteins, it's tetrameric, etc. The diagram to the right shows what other molecules this protein is associated with. Since it's monomeric, there's only one other protein present, but there are 7 chemicals associated with this version in the 3D structure, and these are marked by the diamonds. In the table at the bottom, you can see what these 7 chemicals are. There's 3 sulfates, 3 sodium ions, and 1 calcium ion. The molecular graph window shows a 3D representation of the gene. Click on the Open icon at the bottom, and this opens the structure viewer. Now this is a very powerful tool for visualizing different parts of the enzyme. You can rotate it or increase the size of the view. The little star-like structures are the 3 sulfate ions. The spheres are the sodium and calcium ions. A powerful feature of this software is the ability to visualize specific amino acid sequences. The primary sequence of the protein is shown to the right. Now, if you select certain amino acids in this sequence, the locations in the picture are highlighted. So you can imagine how useful this is if you want to design this protein by changing specific amino acids. The default view is to show the enzyme in a ribbon style, or Richardson diagram. And this is the most common way to depict protein structures in 3D. The coiled ribbons represent alpha helixes, arrows represent beta strands, each are different protein conformations, and the thin tubes are non-repetitive coils of the protein. You can change the format of the structure by going to the styles proteins in order to visualize the protein in a different style. For your assignment, you're to make a copy of 3 different enzyme structures in this exercise using 3 different protein styles. Save these as PNG files. And insert it directly into the Excel table. That's it. Okay, that's it for this assignment. If you have any questions, ask your colleagues or TAs, use Piazza, go to the TA office hours. Good luck with it. Bye.